Hello and welcome. I am Janet Heischetter, and it's my privilege to serve as the Executive Director for the Dystonia Medical Research Foundation, or the DMRF. It is the mission of the foundation to support research that will lead to better, improved treatments and ultimately a cure, while also supporting efforts to increase awareness of dystonia, to educate people about dystonia, and to provide support for those who are affected by dystonia and their families. The DMRF has been serving the dystonia community for nearly 45 years, and we're not going anywhere until there's no longer a need for the foundation's work. The topic for tonight's webinar was suggested by a number of our support group leaders and championed by DMRF Leadership Chair and member of the Board of Directors, Paula Schneider. I want to thank all of our leaders and Paula for the work that they do every day in supporting those who are living with dystonia, and particularly for this program suggestion. Tonight we're going to learn about dystonia genetics and the important role of genetic counselors. Genetics is an area of research that the DMRF has contributed significantly to over the years, and we recognize that it's also a very important topic for patients and their families. And we're fortunate tonight, our speaker, Deborah Raymond, has been a longtime friend of the foundation and just a wonderful resource for the dystonia community. Debbie is a certified genetic counselor, a clinical researcher at the Merkin Department of Neurology Center for Movement Disorders at Mount Sinai Beth Israel Medical Center, where she counsels patients and families about genetic movement disorders and options for genetic testing. She is also a senior coordinator of research studies aimed at understanding the genetic causes of dystonia and Parkinson's disease. We're fortunate to have this dedicated expert with us to help us understand the genetics of dystonia and how genetic counselors can help. It's now my pleasure to turn the program over to Debbie Raymond. Debbie?
Thanks, Janet. So good evening, everyone, and thanks again for joining the webinar. I am so excited to be here tonight to talk to you about dystonia genetics and the role of genetic counseling. So let's start with some review. You all know that our genes, or DNA, make us who we are. They are the instruction manuals for the production of proteins that allow our bodies to function. Genetic mutations are changes in the structure of genes that impair their ability to produce a functional protein. A piece of a gene can be switched or deleted or duplicated where it shouldn't be. Another common name for a gene mutation is a pathogenic variant. Our genes are strung together into structures called chromosomes, like the ones you see in this picture. And our genes and chromosomes come in pairs, one copy coming from each parent. A parent passes one or the other copy on to each child, so there is a 50-50 chance to pass on either copy. In most dystonia, increased risk is associated with having a mutation or alteration in one copy of a gene. This type of inheritance is called autosomal dominant. And importantly, most people with a risk gene for dystonia never develop symptoms. This is called reduced penetrance, which I will explain in a moment. In some rare forms of dystonia, there's a mutation in both copies of a gene, and this type of inheritance is called autosomal recessive. Now, we don't really know the specific reasons why most people with a dystonia risk gene never develop symptoms, but we do know that other genes and environmental factors must play a role. So penetrance refers to the chance that a person with a mutation will develop symptoms. And to illustrate, the jars in this diagram are meant to represent a person's dystonia risk, and the jars fill up over a person's lifetime. You may be born with genetic risk factors for dystonia, and those are represented by the blue marbles, and during your life, you're exposed to different environmental factors that also contribute to your risk, and these are the green marbles. Now, each person has a different risk of dystonia, so each jar represents an individual with a different combination of genetic environmental risk factors. The individuals that are represented by the top two jars do not have enough total risk factors to reach the threshold where symptoms result. That's the most common scenario, while the two individuals on the bottom do. And only these two people represented on the bottom of the diagram will develop symptoms. Now, unfortunately, there's no way to predict which individuals with a particular genetic risk factor will have symptoms and which individuals will not. Another thing that we know about dystonia genes is that we can see varying ages of onset, severity of symptoms, and types of symptoms in individuals who share the exact same gene mutation. So, for example, the gene mutation that usually causes early onset generalized dystonia can sometimes cause adult onset focal dystonia. And mutations in the gene for dopa responsive dystonia can cause early onset dystonia in some people or late onset Parkinson's disease in others. And just as we can't predict whether symptoms will develop, we also can't predict when and what symptoms or how severe they will be. So when we speak with families, we rely heavily on trends and observations that have been reported from studies of families and individuals with dystonia. And this is really an essential way in which dystonia research and your participation in research has benefited dystonia patients. What I love about this table is that it's really a snapshot of how far we have come in understanding dystonia genetics, because here in this column is a list of dystonia genes that were mostly unknown just 20 years ago. And here again, you should feel very good about the part that you have played in helping to identify these genes by participating in and supporting dystonia research. And of course, the DMRF has played a huge role in this. There are a few other things that I want to point out about this table. First of all, you will notice that there are two different categories of dystonia here on the left side. Isolated dystonia is the type where dystonia is the only symptom, while combined dystonia has a second disorder in addition, or a second movement disorder in addition, such as Parkinsonism or myoclonus, and a few forms are also paroxysmal or episodic. The most common genetic test for dystonia will actually look for mutations in the genes in this table. There are also other complex conditions where dystonia can be one feature among a constellation of other types of symptoms, but these types are very different from the ones we're going to be discussing today. So now you're getting a sense of some of the complexities that we know exist in dystonia genetics, and that is a great segue into the next part of our discussion, which is the role of the genetic counselor. Genetic counselors are trained to identify genetic risk in a family and to help individuals and families understand their risks and testing options, as well as how the results of testing may affect them and their families. And this involves drawing and assessing the family tree for trends that are linked to certain genes, recommending appropriate testing, explaining the inheritance pattern in the family and the chance for relatives to develop symptoms, explaining testing options, including what can be learned from testing and what questions may remain, what are the benefits and drawbacks of testing, what are the possible results and what they could mean, how the results might change family planning options, setting proper expectations about the utility of the testing, lending an empathetic ear at times, and acting as a liaison and advocate for patients. There are at least two different ways that someone could use to find a genetic counselor. The first would be to ask your neurologist or your obstetrician if there is someone that they generally refer to or recommend. Another is to go to the website of the National Society of Genetic Counselors and use their Find a Counselor link, and you see that here on the right. You can search by specialty, for example, neurogenetics or pediatric, adult, or prenatal specialties. Now, who should be counseled and tested? So let's start with who should be counseled. And basically, any adult who wants to learn about their personal risk of dystonia can opt to be counseled, and based on that can decide whether to have testing. However, genetic testing in the family should always begin with a symptomatic relative. This is because testing in an unaffected relative is only meaningful if the risk gene in the family is known. If a family risk gene is known, a family member who is tested for that specific gene will end up with a clear yes or no answer about whether they are at risk. Without this information, a negative result may not be a true negative because the risk gene in the family may not have been one of the genes tested or may be unknown. And in fact, people with the most common type of dystonia, which is adult onset focal dystonia, are most likely to have a negative test because while we know genetics play a role in these forms, the genes are still mostly unknown and therefore we can't test for them. So if a patient has a negative test, there's no point to testing the relatives. I also wanted to mention that in general, it's not recommended to test children with no symptoms, and there's a variety of reasons for this. First of all, it is felt that a person should be able to decide when they are mature enough whether they want to know if they carry a dystonia risk gene or not. And secondly, there's the fear that if a parent knows a child's status, they may treat or nurture the child differently than they would have otherwise. So does testing help with treatment? And the answer is yes, particularly in less clear-cut cases. But when a subtype is clear, genetic testing can still give an extra level of certainty. It may also limit the need for additional diagnostic testing and can be important when deciding about deep brain stimulation or DBS based on patient outcomes for the different genetic etiologies. In addition, some mostly rare dystonia is treated based on genetic diagnosis, for example, dopa-responsive dystonia. And for those who want to learn more about gene-based treatments in mostly rare, complex forms of dystonia, the article that is referenced at the bottom of the slide is a good resource. Okay, so what are the benefits of identifying a gene mutation? Finding out the cause of the symptoms and validating your symptoms can be very important for patients, particularly if you're someone who's been told, just stop it, or that's all in your head. And testing may help to understand prognosis based on other reported cases with the same risk factor. Though it's important to remember how different symptoms can be in different people even with the same mutation. Testing can be helpful to medical management, but this may be even more the case in the future, and you'll understand why later on. It can also help to understand inheritance and risk to relatives and can open up options for family planning, including pre-implantation genetic diagnosis, or PGD. And it may also play a role in research participation. And unfortunately, there are also potentially some drawbacks to testing. And those are that you may be left with uncertain results. There may be feelings of guilt or anxiety around passing on a mutation to a child. Some people may worry about telling family or about the reactions of their relatives. And often results will not significantly change management of symptoms. There's also no current prevention or cure. In some cases, there may be reason for concern about insurability, especially vis-a-vis life and long-term care. And some types of testing may be costly. So I want to take a moment now and address the question of insurance. And I'll tell you that there is a federal law called GINA, or the Genetic Non-Discrimination Act, which says that you cannot be denied health insurance based on having a risk gene. And GINA specifically relates to those at risk for disease. Those who are living with a genetic disease are currently offered a similar protection by the ACA, or the Affordable Care Act, which currently protects individuals with pre-existing conditions from being denied medical coverage. However, neither of these laws offer any protections for people applying for life or disability insurance. Now, dystonia does not affect life expectancy, though some people do have significant disability. However, access to those insurances for patients is likely driven more by their symptoms than by their genetic diagnosis. What we don't know is how an otherwise healthy individual is ranked when a risk gene has been identified. Now, insurance concerns are not necessarily a reason not to be tested, but they may weigh into a person's decision about testing. And one option people have is to look into obtaining insurances before initiating testing, although this may be less of a concern for dystonia than for some neurodegenerative conditions, for example. Now, there are a number of different options for testing. And the first one, a dystonia panel, is the best test for looking at known genes and the one that's most often used for isolated and combined dystonia. The advantages of this test are that it can identify the most mutations in these genes. It is the fastest. It takes about 2 to 4 weeks to get results. It's the cheapest, and it is the least likely to have an unclear result. The disadvantages are that you can only test the specific genes on the panel, and these may differ between labs. When we order a panel, another test that we sometimes order is called a microarray, which can pick up additional rarer types of mutations. Whole exome sequencing, or WES, is better if a cause other than the known genes is suspected. And WES is a way to screen all of our 20,000 genes, which is only about 1% of our DNA, but is the most important part for our functioning. And the advantages include the chance of finding a new or unexpected genetic cause of symptoms, and that also the same data can be reanalyzed later on when there's more information known. The disadvantages are that certain types of mutations are not detected. Testing is limited by current knowledge of dystonia genes that is used in data-driven analyses for the results. There's more chance of unclear findings. It's expensive if not covered, and results take about 3 months to come back. The next level is a test called whole genome sequencing, or WGS, which is still primarily a tool used in research, but is definitely coming soon to clinical testing as well. The advantages are that WGS is a way to screen a person's entire genome, so 100% of our DNA, and it can pick up all types of mutations. The disadvantages are that it is also limited by current data used in analysis, as well as by the breadth of data that is generated and current methodologies and analytic tools. There is even more chance of unclear findings. It is the most costly, and it takes the longest to get results. There are a number of possible results from testing, and discussing these prior to the test helps people to prepare for what to expect. A positive result means that a mutation is identified that clearly contributed to causing symptoms. A negative result means that no mutation was identified. And here it is very important to keep in mind that a negative result does not mean that your condition is not genetic. It is still possible that your symptoms resulted from a gene mutation that just could not be identified through the test. An uncertain result happens when a variation in a gene is identified, but there's not enough evidence to determine if it is disease-causing or just a normal difference between people. This added bit of uncertainty can be tough to take at times for people. Unexpected results are only relevant to WES and WGS testing. In these tests, you may learn that you have a mutation in another gene for cancer or heart disease, unless you opt out of receiving this information. Now, when an alteration in a gene or a variant is identified, there are a number of factors that are used to help determine whether it is the cause of disease or not. A variant is more likely to be the cause of disease or pathogenic if it is very rare in the general population, if it has been seen before in symptomatic individuals, and if it is predicted through available evidence to impair the function of the gene. Direct-to-consumer testing, or DTC, is a way to find out some things about genetic risk factors for disease. Its selling points are that it's easy, it's relatively inexpensive, and it allows you to bypass insurance and medical professionals and just get it done yourself. The big question is whether you're getting what you think you're getting from the process, and many concerns have been raised about this. First is the fact that DTC is not ordered by a physician or genetic counselor. This means that there's no guidance to ensure that the appropriate testing for a specific patient is ordered. There's also no pre- or post-test counseling to help patients understand the implications of testing and to help anticipate and interpret results. DTC is also limited in scope and in breadth because it only includes certain genes and only certain mutations in those genes. DTC may not be less costly either, and in fact, a more comprehensive dystonia panel often costs less. This testing also is different in that it includes certain genes related to other disorders such as Alzheimer's, Parkinson's disease, cancer, and not everyone wants this information. Unfortunately, the reports may also be confusing or hard to interpret, and there is a risk that the results may be misinterpreted. Finally, a person may experience unexpected emotions and not have anyone at hand to contact for help and information. So as you know, one of the benefits of genetic testing is the option to use the information to prevent a child from inheriting a risk gene. One approach for doing this is called pre-implantation genetic diagnosis, or PGD. In PGD, in vitro fertilization techniques are used and fertilization occurs in a laboratory. When the embryo is just a ball of cells, 1 or 2 cells are drawn out and analyzed for the risk variants, and only the embryos that are free of the variants are implanted into the mother. Now, the biggest downsides to this process are the cost, which may or may not be covered by insurance, the chance that the process may not result in a viable pregnancy, and the potential emotional toll of the process. Now, I'm going to leave you with some thoughts about the future of dystonia treatment. In general, the hope for dystonia, as well as many other chronic diseases, is that we will move from symptomatic therapy to therapies or cures that treat the underlying genetic cause of symptoms. While for the most part this is not being done yet for dystonia, a number of gene-based trials or treatments are being developed for other diseases, and much of what is learned could be used in dystonia as well. Now, there are a few different avenues for these treatments. Depending on the problem caused by the risk gene, a pharmaceutical could be used to target and eliminate a toxic protein. to augment or support the action of the residual protein that is produced, or to bypass the problem entirely by replacing a missing protein in a biochemical pathway. And these are illustrated in this figure in points 2 through 4. A second avenue involves inserting a working gene into the cells. This process is called gene therapy, and that is option 1 in the figure. Each of these processes have many challenges, but a lot of headway is being made. And so while these new treatments will likely take some time, there is more hope than ever before that we will get there. So that is the end of the presentation, and I'm going to hand it back over to the DMRF for the Q&A session.
Great, thank you, Debbie. That was terrific, complex topic, and you really helped us to understand it. We got a number of questions. So starting with, we have a mother who has dystonia, didn't specify what kind, but we actually get this question in the office quite a bit. There's an adult in the family who's been diagnosed with dystonia. They have not yet been genetically tested, and their kids want to know what the risk is for them developing this dystonia. So what advice would you give this family?
So basically is one of the questions would be whether there's anybody else in the family with symptoms, and also what type of symptoms the mother has, at what age did they start, and what part of the body is affected, and are there any other symptoms besides dystonia. In general, I would hope that the mother sees a movement disorder specialist who can also help to understand the type of dystonia, and then to work with the doctor and genetic counselor to understand the risk to the children better through genetic counseling and testing.
Right. And you mentioned that with a lot of adult focal onset dystonias, the genetic testing may be negative because we don't have specific genes at this point.
Correct.
Right.
We can say some general things, however, because we know that dystonia is usually inherited in an autosomal dominant pattern with reduced penetrance. So we can say that if the mom has a risk gene for dystonia, there would be a 50-50 chance that any of her children would inherit that risk gene, but that depending on the gene involved, there would be a certain risk of them having symptoms, which would be, quite low, maybe 5 to 10% if it was focal dystonia versus maybe about 30% if it was early onset generalized dystonia. So it depends on the gene. It very much depends on the gene and the type of symptoms.
Right, right. We have a number of questions around sort of that direct-to-consumer testing, things like 23andMe and others. First of all, if you could just, again, talk about the reliability or what we know about the reliability of those types of genetic testing. And then do those results become a part of somebody's medical records?
So, good questions. In terms of reliability, so if the direct-to-consumer test is testing for the gene that runs in your family, that is really the information that would be most important for you to know in terms of testing. So it could be, it could give you the right information if you are someone who has the specific gene that they test for in your family. If you don't, and you're looking to find out in general your dystonia risk, then the problem is that they may not test for other genes, and the information that they provide on their website may be either unclear or misleading regarding, what your residual risk is if your test is negative, basically.
Mm-hmm. And do you know if these are become a part of somebody's medical records?
So basically, unless you report that information to your doctor, it would not become part of your medical record. But the minute you discuss it with a medical health professional, you run the risk that that information will be incorporated into your medical record. Okay.
Yeah. And you talked a little bit about penetrance. And can you explain a little bit more about what that is and what we know about why some people who have a gene develop symptoms and why others who have a mutated gene don't develop symptoms?
Yeah, so there's a lot that is not known about that, and it's been the focus of a lot of study, but it's very, very difficult to flesh out, really. We know that having a risk gene increases your chances of having dystonia by a certain amount, but that is not enough to cause symptoms. So what are the other factors that cause symptoms? They could be either other genetic factors or other environmental factors that all interact together to cause somebody to have symptoms. And unfortunately, we don't know what those factors are. So we'd love to be able to tell somebody, exercise or, avoid certain foods or something like that and help them to change their risk. But we don't have that information because we don't know what these factors are.
Great. It points to, again, just the need for more research on this. You talked a little bit about when to and when not to genetically test kids in a family. And we have a question specifically about an older child developing a dystonia, and this parent is wondering if they should have the younger children tested. Could you just sort of talk about that again?
Yeah. So, if the older child has been tested and the gene has been identified, then potentially that facilitates testing in other family members. However, it's not generally recommended to test younger children or children who are too young to choose testing for themselves, because it is felt that that child should be able to choose that when they are old enough to decide that they want that information. And also because it's not clear whether having that information available in the family would change the family dynamics in any way, or, for example, would a parent who knows that their child carries the DYT1 gene try to dissuade them, for example, from being a surgeon or, something like that. So that's kind of an extreme example, but those are the kinds of things that we think about in terms of testing children.
Right. Yeah. We've had a number of questions about whether or not there's a genetic test for cervical dystonia specifically.
Mm-hmm.
Is there a test for cervical dystonia, and how would people go about being tested?
Yeah, so cervical dystonia is one of those that has been very, very difficult to identify the genes for, partly because it's so common and probably very heterogeneous in terms of its causes. And because the penetrance is so low, it's hard to study families with many people affected because those families don't exist. And that's traditionally the way in which genes can be identified or have been identified. So in terms of the testing that's available, there are probably only about 2 genes that we know that are more likely to cause an adult onset cervical type of dystonia. And those 2 genes account for a very small percentage of cervical dystonia overall. So that's why I said that most people who have the available dystonia testing will likely test negative. Now, somebody might ask about some of the other genomic tests that I mentioned, like whole exome or whole genome sequencing. And unfortunately, it's very unlikely that those tests would pick up a new gene for cervical dystonia. It's not impossible, but it's very unlikely that that would be the case, for the same reasons that it's been difficult to find the genes through research studies. There's just not these analyses are very, as I mentioned, very much driven by current knowledge about the genes. So so it would be unlikely that those would add anything to the usual testing that's been done, that is done.
Mm-hmm. A question from someone who has siblings with different forms of dystonia. They have cervical dystonia. They have a sibling with blepharospasm, and as they look back, they suspect that grandparents probably also had dystonia. Should this family be tested?
I'm assuming that those are both cases where the onset happened late.
I think that's a good assumption, yeah.
Yeah, yeah. And so those, it's really not just the cervical dystonias, but really all of the, or most of the later onset dystonias that are less likely to have a positive test. But there may be a small number that would be positive. So it is certainly possible to do the test to rule out the known genes, but in all likelihood, it would likely be negative.
Yeah. We have questions from people who would like to be tested and think maybe based on information shared tonight it would be good for them to be tested, but their doctor's not necessarily supportive of this. So how do people go about getting genetically tested? Where do they go? How does it happen? What should they do?
Yeah. So I would probably recommend, if somebody wants to be tested and they aren't able to do it through their doctor, then they might want to try to find a genetic counselor through the National Society of Genetic Counselors and use their Find a Genetic Counselor feature on their website. That's one option. The other option might be to speak with another one of their medical professionals and see if any of those people have any recommendations about people that they refer to.
Mm-hmm. And just we'll email this out to everybody when we email the link for the recording. But we had a question to have that site repeated. So the website for National Society of Genetic Counselors is www.n as in Nancy, S as in Susan, G good, C carry.org. www.ncgc.org. And again, we'll email that out to everybody with the link to this recording. So thank you for that question. What is pharmacogenetic testing?
So in traditional pharmacogenetic testing has to do with looking for genes that may be associated with a person's response to medicines, and it's used a lot of times in very specific types of conditions, but there are there's a lot of buzz around pharmacogenetic testing, and some doctors do use it. I don't think that it's used very often in movement disorders right now, but for example, psychiatrists sometimes use it.
Right, and ours is just not there yet, right? The science is just not there yet for dystonia.
So. In terms of determining whether you would respond to a certain medicine, no, it's not.
Right. And we have a question of a family that has had genetic testing, it's positive, and they wondered if the dystonia ever, do we know anything about ever skipping a generation?
Yes. Is the short answer. Basically, when people see families that have, say, a parent has dystonia and then the child will not, but the grandchild will, you kind of think of it as skipping a generation. But in fact, what you're seeing is a reduced penetrance. So that gene, that risk gene, was passed on from the grandparent to the parent to the grandchild, but because of reduced penetrance, the parent did not have symptoms. So it's not that the gene isn't there, has somehow skipped the gene has skipped a generation. The gene is there, but the condition is just not manifested because of reduced penetrance.
Mm-hmm. Right. Right. So we have a couple questions about myoclonus dystonia and the SCGE mutation. Might it be possible for someone to have that mutation and have the symptoms of anxiety and those kinds of things, but not have the movement disorder symptoms?
It's probably possible to have that. Yes. Yes. Those symptoms are associated with having a mutation in this gene, and occasionally people may have just those symptoms without the movement disorder.
Mm-hmm. And then this is a little bit of an unfair question, but I think it's one that we get here, and we've had a number of people ask it tonight, and that's around insurance coverage for genetic testing and the cost of genetic testing. Some insurance companies are a little bit better than others about covering this. Do you have advice around what we as a community might be able to do to address these kinds of issues and getting genetic testing more readily covered by insurance companies?
That's a good question. I mean, I have, in my experience, actually, I haven't had too much trouble getting genetic testing covered. Sometimes the more costly types of tests are more of a challenge, but some of it has to do with choosing I have to say, choosing a lab. You know, maybe if one lab doesn't work out with the insurance, to try a different lab that has a different policy or has sometimes a lot of the testing labs also have teams of people who work with the insurance companies and help to get testing covered. So that can sometimes be helpful too.
Mm-hmm.
But in general, I have to say that I have not had too much trouble, for the most part, in the last 2 years or so in getting testing coverage, and that's because the cost of testing has come down so much. You know, years ago, you know, people would get these quotes of thousands of dollars for testing, and that's just not the case anymore. So that's one wonderful thing that's happened.
Mm-hmm. Yeah, yeah. Well, it's good news that you're not having challenges, because maybe that will, you know, spread as well, and more people will have that experience. You talked about the time involved with whole exome sequencing and whole genome sequencing taking quite a bit of time. Why does it take so long?
It's because it's basically a very involved process, both in terms of the methodology as well as the analysis. It's a very very complicated process, and it just takes a long time.
Yeah, yeah. And we want to get it right? So we want them to, yeah.
Exactly.
We've had a couple of questions about what we know about the relationship between Parkinson's disease and dystonia. You know, if somebody has a genetic dystonia, are they predisposed to developing a Parkinson's and vice versa?
In general, not, but there are specific subtypes of dystonia, for example, dopa-responsive dystonia, where there may be an increased chance of Parkinson's, you know, as well as dystonia, or that Parkinson's in dopa-responsive dystonia is actually an alternate phenotype to early onset dystonia. So in those rare types of dystonia, there is an association with Parkinson's disease, but in general, there is no known relationship between, you know, most of the dystonia genes and Parkinson's.
Mm-hmm. So we have a question from someone who wanted to know if they were doing one of the direct-to-consumer testing, like 23andMe, would they be able to take that to a genetic counselor to help them interpret the results?
Yes, you can do that.
Okay. And then
you would have to. Sorry to interrupt. You would have
Sure.
to log in. You would have to log into your account in the genetic counselor's office because there's no printed report. So you'd have to actually look at it. Have them look at it online.
Mm-hmm. Right. Yeah, that's good to know. We always welcome this question. Someone wants to know how they might be able to find out about studies that they might be able to be eligible to participate in and advance our understanding of genetics and dystonia.
So I would direct those people to you. The DMRF is probably the best resource for directing people to studies. Let's see, there are a number of well-known studies as well, including the Dystonia Coalition study, and there's, you know, once there are trials for dystonia or new trials for dystonia, you can also keep track of those on a site called clinicaltrials.gov, and
Right.
those are research into potential therapies. Those
Right.
types of trials.
Yeah, and thanks for the plug. I'll just respond to that as well. The DMRF does try to let everybody know when there are studies that we've been made aware of that people can enroll in, and we'll send out the clinicalstudies.gov site with the link and the information on the National Society of Genetic Counselors as well, so everybody has it. But we encourage people to participate in the studies that they're eligible for, because as Debbie said, that's how we're able to learn new things and move things forward, which is really important. So thank you for the question. And that's really it. That's what the time we have for questions tonight. And those that we didn't get to, we'll make sure that we address in future issues of the Dystonia Dialogue. And I want to thank Debbie for your presentation and answering our questions, and just for the work that you do every day in supporting the dystonia community. You are a treasure, and we are fortunate to have you in this community and working on behalf of those who are living with dystonia and their families. So thank you very much for your work and for the presentation tonight.
Oh, you're most welcome, and thank you for those kind words as well.
Well,
say the
so
same
thank you.
About you.
Well, thank you. We're honored to serve this community. So thank you, everybody. We wish you all a good night. And again, thank you for tuning in tonight. Good night.